Hello everyone, my name's Catherine, this is Emily, and Chris. We're going to present the lab module we did for microsatellite DNA. Alright, we are humans made of cells that contain nuclei. These nuclei contain 23 pairs of chromosomes, and the word locus refers to a specific location on a chromosome. Now, a chromosome is made up of a protein in a single DNA molecule. In this lab, we talked about a specific type of DNA called satellite DNA, which is composed of short repeating DNA sequences. They're usually located either in the centromeres, which are the central part of the chromosome, or the telomeres, which are the very ends. And satellite DNA does not code for proteins. We're still trying to figure out exactly what it does. More specifically, we talked about microsatellite DNA, also known as short tandem repeats, which have repeating units of 1 to 6 base pairs. Now the big question, why do we care about microsatellite DNA? Took me a while to figure this out, but I've come to the conclusion that the important part is that the number of repeating units, expressed as the allele number, is the basis for human variation, and that's why it's so important. The purpose of the lab we performed was to use genetic markers at 10 loci to identify humans. The first process we performed was DNA extraction. Like this fine gentleman you'll see on the screen, we delicately removed our epithelial cheek cells, and then we used magnetic beads in the magnetic sand to separate the DNA to the bottom of the flask. No. The second process we performed was DNA purification. We used 3 different types of buffers. The first one was the lysis buffer, which disrupted the cell membrane and cleaved the DNA. Then we used a wash buffer, which removed the lipids in the proteins from the DNA. And then we used an elution buffer, which released the DNA from the beads. Next in the lab.
All right. So the way that we did this was through a process known as PCR, which is the polymerase chain reaction. And basically what this is just a way to replicate DNA outside of the cell. And so there are 2 parts to the DNA double helix that are important. There's the template strand and the coding strand. And the coding strand is the one that we care about the most because it's the one that gives us the proteins and the one that helps with phenotype. So in order to get the coding strand, we use PCR on the template strand through a process called multiplexing. And multiplexing is when you replicate multiple loci in one reaction. So it's much more efficient, but it does require that each loci has a different pair of markers. So how does the process of PCR work? First, you take your DNA molecule that you have, and you heat it to 90 degrees Celsius, and then cool it back down to 60 degrees Celsius. This allows all of the primers to bond to the template strand. Then the reaction is warmed again to 72 degrees Celsius, so that DNA polymerase can synthesize the coding strand using DNTPs, which are basically all of the base pairs in the DNA. So why is PCR important? Well, it allows us to get multiple DNA samples from a very small sample, which can help in determining parentage, in forensics, and in wildlife conservation. So the way that we did this in the lab, it was in 2 different parts. So the first part was through gel electrophoresis. So gel electrophoresis was a 3-locus multiplex PCR. And so after extracting the DNA in the process that Catherine explained, we inserted it into a gel matrix. And then through electricity, the negative phosphate groups in the DNA were able to travel towards the positive electrode. And all of these DNA fragments were different lengths depending on which fragment they were, so they all traveled different distances. So we got a picture like you see up there. And then using a template, we were able to compare our results to that template in order to determine which DNA fragments were which. The other way that we did this was also the electrophoregram. And so the electrophoregram is a 10-locus multiplex PCR, and it results in a chromatogram, which are the results that you see with all of the different colored peaks for where your alleles were, or your loci were. And the peaks show your genotype. So if you had 2 peaks in one loci, that meant you were heterozygous, and 1 meant you were homozygous. So what does that mean? Heterozygosity means that you have 2 of the 2 different alleles, like the brown and the blue M&M, and homozygous meant that you have 2 of the same, like the 2 brown M&Ms. So through these, we were able to find our own frequencies and the probability that they would occur in nature again, using the gene reference tables in the back of our book, as well as the Hardy-Weinberg equation.
All right, so what Hardy and Weimer recognized was that there's, within any population, that there's two types of alleles. You have the dominant allele and you have the recessive allele. And that since you only have two alleles, that the frequencies of those alleles must add up to all the possible outcomes in that population. So therefore, they derived that equation, P plus Q equals 1. Following from that, they, in conjunction with the Punnett square, they figured out how those alleles interact within the population to form those four possible outcomes in the square. And that's how they got the well-known P squared plus 2PQ equals Q squared. Now, whenever you're dealing with the Hardy-Weinberg equation, you must take into account the assumptions that they used when creating the equation. Basically, mating is completely random, the population size is infinite, there's no gene flow in and out of the population, mutations are not occurring, and then you don't have natural selection, essentially meaning that you don't have another genotype coming in that's going to be better for survival compared to another genotype. So why bother? So why is this important? Basically what the Hardy-Weinberg equation does is it tells us whether a population is in equilibrium or not. If the population is not in equilibrium, examination of the particular frequency for that genotype in a population can tell us actually what conditions are not satisfied. In conclusion, what they found out was that recessive alleles in a population are very unlikely to go extinct because, such as blonde hair and blue eyes, because not enough conditions were violated in order for that to happen. Meaning that blonde hair is essentially not naturally selected over darker hair for better survival. Thank you.